Bacteria reproduce very simply and rapidly by doubling their contents and splitting in two. Just one bacterium, dividing every 20 minutes, could produce nearly 5,000 billion bacteria in one day. Bacteria reproduce very simply and rapidly by doubling their contents and splitting in two. Just one bacterium, dividing every 20 minutes, could produce nearly 5,000 billion bacteria in one day.
In this video, we're going to explore binary fission, which is the process by which prokaryotic organisms, such as bacteria, divide and reproduce. Binary fission is the equivalent of the cell cycle, but the two are very different, and the cell cycle only occurs in eukaryotic organisms. To understand binary fission, let's look at a simplified version of our bacteria, to better show the bits that you need to understand. Bacteria have both a large circular piece of DNA, which contains the important genes that they need for basic survival, plus extra smaller circles of DNA, called plasmids. When the bacteria wants to divide, the cell firstly replicates all of its genetic material. The cell then grows, and the two large circular strands move to opposite poles of the cell. However, the smaller plasmids are more randomly distributed. Finally, the cytoplasm itself begins to divide, and a new cell membrane and cell wall form around the new cells. Here, we've shown the wall and membrane as a single line. But don't forget that in reality, there are two layers: the cell wall on the outside, and the cell membrane on the inside. As binary fission is such a simple process, prokaryotes can divide very rapidly, given the right conditions, which generally means a warm environment and plenty of nutrients. For example, some bacteria can divide every 20 minutes, which means that a single bacteria could form a colony of over 250,000 in just 6 hours. In your exam, you might be asked to calculate something like this for yourself. For example, a bacterial cell has a mean division time of 30 minutes. How many cells would it produce after 3 hours? First, we need to find how many division cycles there has been, by dividing the total time, which is 3 hours, or 180 minutes, by the mean division time of 30 minutes. In our case, this equals 6, so we can tell that 6 divisions will have taken place. To calculate how many cells that makes, we will take our original cell, of which there is only one, and multiply it by 2, 6 times, which in total gives us 64, which is our answer. To write this multiplication out more simply though, we could just write 2 to the power of 6. It's important to remember that each of these 64 bacterial cells would be its own organism. This is in contrast to the cell cycle, where new cells are still part of the same overall organism. And that's it for now. If you enjoyed it, then please give us a like and subscribe, and we'll see you next time.
Bacteria reproduce very simply and rapidly by doubling their contents and splitting in two. Just one bacterium, dividing every 20 minutes, could produce nearly 5,000 billion bacteria in one day.
Phases of bacterial growth with scientist Cindy. When we analyze bacterial growth, it is convenient to create a graph of the number of bacterial cells in our culture versus time. This type of graph is called a growth curve. Here is just an example of one possible growth curve. Keep in mind that the time in which all of these phases will be experienced will vary depending on the amount of available space and available nutrition and the type of bacteria we are growing in the cell culture. All bacteria growth curves grown in cell culture in the laboratory exhibit the same four following phases: the lag phase, the log phase or exponential phase, the stationary phase, and the death phase. The lag phase of the growth curve is when we have no active growth, so the population of cells remains the same during the lag phase. This is because the bacteria are adjusting to the new environment. The cells will not undergo binary fission until they have adapted to their new habitat. Once they have adapted, we will experience the log phase or exponential phase of bacterial growth. Here, there is plenty of nutrients and plenty of space for growth to occur. This exponential growth, or logarithmic growth, will eventually level off. This leveling off is due to the population of bacteria beginning to run out of available space and available nutrients. Also, the cells will experience a continued buildup of toxins. Due to these factors, the log or exponential phase will give way to the stationary phase. In the stationary phase, we see that there is no real change in the number of bacteria in the cell culture. In the stationary phase, the number of bacteria cells in our cell culture remain fairly constant due to the fact that the number of cells being created will roughly be equal to the number of cells dying in our cell culture. After some time, the depletion of nutrition and space, along with the buildup of toxins in our cell culture, will cause more cells to be dying than cells that are being produced. This gives us our death phase of the bacterial growth curve. After some time in these conditions, we will eventually see that all of our cells in the cell culture will die.
Welcome back, friends. Let's talk a little bit about chemostat. Now, chemostat, as you all know, is a machinery which is used for maintaining a continuous culture. Now, for understanding chemostat, you must know what is a continuous culture, right? Continuous culture is a culture process or a culture technique where we need to culture a bacterial load continuously throughout the time, right? That's why it is called continuous culture, because actually there are majorly two different types of culture. Major two types, actually three types, but majorly two types. One is a batch culture, where we are having a batch system, that is we are having a culture vessel where we are having all the nutrients and we put our inoculum and we let it grow at the normal at their optimum temperature for a particular period of time. That's a batch culture process. But sometimes what you need to know, sometimes we need to culture that microorganism, mostly bacteria, in a continuous manner, so that they continuously grow and divide, and then we can elute the content of microbial cells out of that vessel, right? So we need to do those things. So for that reason, this is the process called this is the machinery called chemostat that we hire, that we use to maintain and balance that continuous culture process. So what we are having nothing, but it's not a very fascinating machine. It's a very simple drawing. It is a vessel, culture vessel, just like the batch culture, but definitely for continuously maintaining a culture, we need to have continuous supplement for nutrients, obviously medium, which is supplied by medium. Fresh medium means fresh nutrients for the growth of bacteria, and also bacteria can produce many excretory materials, many bad materials, toxic materials for them. So we need to excrete that out. We need to take that out of the medium too, otherwise they cannot grow, otherwise the growth of bacteria may slow. So for that reason we need to exclude them out, as well as once the culture are dead, once the bacteria are done with it, and once they are producing our product, because most of the time these processes of batch culture or continuous culture are taken in industrial sectors, where we use bacteria or other microorganism, and we put some substrate in, and we allow that microorganism to convert that substrate into product, right? So here comes the role of bacteria in industry. So once the product is being made, we need to elute that product out of this whole container, and that should be done if we maintain the constant rate of the conversion of substrate into product. We can do it in the batch way, that once the product conversion is done, then we stop the reaction, we take out it and we filter it, we get the product and we set up a new batch. But for continuous culture, we continuously do these tasks throughout the time. Now for that reason, remember two important things. One is the fresh medium input as well as fresh oxygen input, because if the process is aerobic, which demands a lot of oxygen to be mixed with the media, we need to supply the oxygen. So both these things, medium as well as gas, fresh air, we need to supply them all together. So two imports are required and one exporting region to take out the eluent containing the microbial cells, right? So we need to take that too. So we need to have these two different sections extra than the batch culture. Batch culture is completely closed, but in this case we can see two imports and one export system is there. So imports are actually one for fresh medium reservoir, and it have a flow rate regulator, because if the flow rate is too much higher, then it may jam this, it may overflow it, right? So for that we need to have a flow rate regulator there, and we need to have a air or other gas insertion, right? So here we are having sterile air or gas insertion device. It's nothing but tunnel like structure, but it is definitely sterile. Sterility is the biggest factor to maintain a chemostat, because there are openings, right? So once there are openings, there is a chance of air coming in and out from other regions, or water coming in and out from outside, which may contaminate the materials. So for preventing that, we need to have a tight regulation of these openings, right? And we are actually doing so. And on the other hand, we have the excretion system, where we are having this tunnel like structure, again another pipe through which we are having the overflow of all those nutrient materials coming out, right? So this is the schematic representation of the whole continuous culture device. So chemostat is nothing but a continuous culture device, right? Maintaining a culture inside that huge chamber, right? So that's the whole process of chemostat, and how we use this chemostat for completing the continuous culture system, right? Or for maintaining a continuous culture. So that's it guys, and I hope that's helpful. Thank you.